Today, we're going to talk about stem cells and how they can be potentially used as a treatment for Huntington's disease. Stem cells are special kinds of cells that have the ability to turn into almost any type of cell in the body. Think of them like blank pieces of paper ready to be folded into anything, like a paper airplane, a boat, or a bird. Scientists have developed methods of turning stem cells into a heart cell, a muscle cell, or even a brain cell. This enables scientists to ask questions and do research about people's brains in health and disease. Since HD is a neurodegenerative condition where brain cells are lost over time, one potential use of stem cells could be replacing the lost cells using a stem cell transplant. Several groups of researchers are working towards this goal of advancing stem cell transplants as a therapy for HD, but key questions remain. What would happen to the new transplanted cells? Would they turn into cells that are sick in HD as well? A recent study published by Dr. Elena Cattaneo and her team from the University of Milan answered some of these questions. In this study, they sought to understand what effect cells with HD had on cells without HD. In the lab, stem cells are grown in a flat dish, but we know that the brain is not 2D. So scientists have developed advanced techniques allowing brain cells to be grown in 3D structures. These structures are called organoids, or mini-brains. While mini-brains can adopt some of the cellular features of a brain, like connections between cells, they don't have the ability to transmit thoughts and feelings. From the outside, they look like pinkish snot, but they are incredibly complex behind the scenes. The brain cells form connections and send signals to each other, enabling researchers to understand how HD affects connections and communication between cells. Brain cells communicate by sending signals across a tiny gap called a synapse. This is where the tips of two brain cells reach out and send information to each other across this tiny gap. In HD, this flow of information is reduced over time as the disease progresses. Dr. Cattaneo's team saw the same scenario in HD mini-brains, that there was less communication at the synapse when compared to mini-brains that don't have the HD gene. But what happens to cells that don't have the HD gene when they are combined with cells that have the HD gene? Dr. Cattaneo's team used mini-brains to model this process. When the HD and non-HD cells were combined, the whole complex resembled the mini-brains without the HD gene rather than with HD. In fact, the cells without the HD gene had a positive influence on those with the HD gene. The communication between cells across those tiny gaps called synapses was also improved, suggesting that the cells without HD might be helping the cells with HD to communicate better. While things were improved, some cells still died in the mixed mini-brains, suggesting that while cells without HD help, they can't completely protect cells that have HD. Mini-brains sound really cool, but there are a few limitations to keep in mind. It is important to remember that these are cells grown into clumps in a dish and don't completely reflect a human brain inside a living person. The mini-brains were created with cells with or without the gene for HD, and so were mixed populations from the beginning. However, in people with HD, the cells or stem cell treatment would be added after the person had a fully formed brain. The human brain is incredibly complex, so these studies are important to help us understand how HD affects cellular and molecular features in brain cells. This knowledge helps scientists to develop treatments and move therapies like stem cell transplants forward.